Good morning, dear students. I hope you are in a good situation, and I hope to come back in a short time to the campus. Today, I will talk about the new lectures about the mutation. I think you are all here on the Zoom. Tell me your names. Diana?
نعم.
Yes.
Yes.
Zardesht? Zardesht, where are you?
Yes, دكتور.
Gosh. Maryam?
بلى.
Where are the others?
دكتور أوجد موجودة.
Juan? Juan? Dania?
نعم دكتور.
Jiman?
بلى سيدة.
Zainab?
بله دكتور.
Solar?
صورت دنيا دكتور.
شو؟ Mute your mic. Solar?
دكتور صورت دنيا.
Okay. Lana?
بلى دكتور.
Helene? Yahya?
بله دكتور.
Yusra?
بلى دكتور.
Helene? Absent? Salah
بلى دكتور.
absent? Huh?
يا حاضرم دكتور.
Helene?
أسهلينم.
Tamam.
أس يا حاضرم.
Okay.
درست.
Solar? Absent here. And the Juan?
بلى دكتور.
Okay. So that you're all here. The outlines of my lectures. Introduction. What are the mutation or the meaning of a mutation? We have somatic mutation. We have germ layer or sex mutations, types of mutations at the gene level, and DNA repair systems. Because this lecture is long and we have a short time, about 40 minutes, so that I will give you, or maybe give you the one part of this lectures. Now, if you watch these pictures here about the two strands of the DNA, and after replication, one of these strands become a mutant and the others the original or origin, so that here the mutation will start. Now, the introduction. The flow of information within a cell involves the transcription of DNA to messenger RNA and the translation of messenger RNA to protein. You know, this is, or this steps will start with the cell or the nucleus, exactly in the nucleus. The messenger RNA take copy of the DNA and then travel to the cytoplasm, to the ribosomes, to translate this messenger RNA to the protein. The flow of information, information between generation involves DNA replication and distribution to two daughter cells. I mean here, if anything's happened in the translation, maybe this wrong happens, maybe followed to the two daughter cells during the division or during cell division. Therefore, any change in DNA that it is replicated and passed on to the future generation may affect protein structure and function in the next generation, so that anything's happen wrong or any wrong happen in the replicating the DNA, it be effect on the protein in the next generation or offspring. Also here, DNA replication is highly occurrent process, meaning this process is very immune process, so that any mistake happen, it be affect in the next generation. So that here, the replication of highly occurrent process, if this happen, but mistakes can occasionally occur. It's not always, occasionally, and sometimes, such as DNA polymerase insert the wrong base. DNA polymerase, you know this enzyme, putting or this working to put the correct nucleotide in the correct place, so that any wrong happen in the DNA polymerase may be affect on the protein consistent in the next generation. Uncorrected mistake, if the mistake it happened, may sometimes lead to serious consequences such as cancer. Cancer here meaning any changing in the sequence of the DNA maybe lead to the cancers. Repairing mechanisms correct the mistake. We have mechanism for repairing. This is also doing by DNA polymerase, so that repair mechanisms correct the mistakes. And sometimes in rare cases, meaning in very few cases, mistakes are not corrected, and lead to mutations. In other cases, repairing enzymes are themselves a mutant or disactive. You understand, or I think you understand, that the DNA polymerase correct any mistake happen in the DNA. And sometimes, or in rare cases, in very few cases, maybe the correct happen, and the DNA polymerase cannot repair this mistake, or the enzyme themselves, it be a mutant or deactive. Maybe it's happened. Here you can see in this picture normal gene and a mutant gene. So that if you see my pointer, can see here, this is in a brown color. Here, it be happened, this has happened, the mutant or put incorrect nucleotide here so that it make other type of protein not require the protein. What is the mutation? The term mutation refers to alternation in the nucleotides, meaning they have some changing in the nucleotide sequences of the DNA that make up a gene. The mutation may be harmful. I think most of the mutation may be harmful, and in very few cases, it be beneficial or neutral, meaning neutral don't make any effect or any changing in the protein, but most of them harmful. The mutation may occur in somatic cells. As I told you before, maybe happen in the somatic cells or in the sex cells. Here in somatic cells and be passed to overspring. Overspring, the meaning here, the generation or the next generation mean here. Or may occurs in gametes. Here, I mean the sex cells, so that the mutation may be happen in somatic cells or in the gametes or sex cells and pass to overspring on inherited. You understand inherited, wirathi, inherited to the springs or offsprings. Here, the DNA during replication we have a mutation here in this two nucleotides. The first one here, the first nucleotide or the first pair, GC, but here in the mutation, GT. And you know that the adenine and guanine, it's purine, a purine ring, and thiamine or cytosine, pyrimidine ring. Here, this is make the mutation, and consequently, it be produced a new or unfavorite protein, and this is called a mutation. About the somatic mutations. Somatic mutations are mutations that arise in somatic cells, as I told you before. They are frequently caused by environmental factors, such as exposure to ultraviolet radiation. Any radiation make change or make the mutation in the somatic body or somatic cells. Also, exposed to the chemicals also make the mutation, or uranium, as it happened now in the world. The mutant cells are passed to the progene, meaning offspring or a new generation, through the process of cell division, so that if this has happened for the parent, it be passed to the offspring during cell division. The effect of these mutations depend on the cell type, so that here, important thing, cell type. Here, cell type. This is the first thing, in which they occur and the developmental stage. This is the developmental stage of the organism. Meaning here, this organism in the early stage of their life or in the it's old. This is also important, so that the type of the cell and the developmental stages. This is effect on the mutation. If this mutation occurs early in development, the clone here meaning any mixed or any changed happen in the DNA, the clone of the mutant cell will be larger. So that in the early, he mixed it in the red color, early, it make large change in the somatic mutation, and the versus, if it's happen in the late in the development or old development, this mutant cells will be few, not large. Somatic mutation can give rise in various diseases, including some types of skin cancers and leukemia. These two important examples about somatic mutations. In this picture, you can see here in the early somatic mutation and the other later somatic mutations. Here we can see here sperm and egg, and this has happened here, the mutation, and this is a mutation, it'd be long. This is not, sorry, this is not, this happened in the somatic part, but if don't confuse, I mean here the somatic or the changing in the DNA or in the protein happens. It starts with the zygote and then divisions to a lot of cells, then become the whole human. Here you can see here the mutation it be in the early, this mutation it be larger or great. In the other side, later somatic mutations it be very small or not be affected or not make a very big problem. Germline mutation. Here I mean about the sex cells or gametes. These occur in the cells that produce gametes. If a mutant sex cell share in fertilization, then the mutation will be passed to the next generation. It's simple. Any mutant happens in the germ cells, these will be passed or this mutation will be passed to the next generation or the offspring. Maybe an individual who has a normal phenotype can hold undetected mutant sex cell. Here, I mean, sometimes the person holds some mutant, but it does not appear detected. You can, or not detected them. Hold the mutant, but not detected him. They have a mutation, or the mutation will be happened, but no anyone can know that. This mutation can be detected only where or when, only if they are included in a zygote. If you have zygote, you can, from the zygote, you know this happened, or did this happen in the egg or in the sperm. There are diseases called hemophilia. This is hemophilia in the European royal families. عوائل الملكية في أوروبا. They have these hemophilia diseases, so that it's related with the X chromosome. X chromosome, one of the sex chromosome of the human. X chromosome linked hemophilia, a mutation in European royal families, is thought to have arisen. It be start in the germ cells of a Queen Victoria. From the Victoria, this is all that happened. Victoria of the royal of England, or one of her parents. This mutation was expressed only in her family males, so that this disease, hemophilia, can be detected only in the males of the royal family. It not be detected in the females of the royal families. So that this is example about the mutation in the germ cells. Here, general mutations, maybe the mutation in the sperm, or maybe the mutation in the egg happen. So that this is the zygote, and this is passed to the offspring or the new generation. And some of them here have this mutation, and the others hold the mutation, but it's not detected. The causes of mutation. DNA failed to copy accurately. When a cell division, it make a copy of its DNA, and occasionally the copy is not quite perfect due to exposed to any environmental factors. This is because the DNA polymerase, as I mentioned before, initially make about 1 in 10,000 base pairs error. The error may be happen, but this error is very, very little. maybe 1 to 10,000 base pairing. This is if happen, it's normal. Sometimes enzymes or always the enzyme or polymerase, DNA polymerase, a proofread إعادة قراءة read the DNA and correct these mistakes. Come back and correct the mistakes if they happen. The new error rate for the DNA that has been a proofread is 1 in a billion base pairing error. To more understand this sentences, I mean during the translation, maybe 1 mistake happens for 10,000 base pair. Maybe 1 mistake or 1 error nucleotide putting for 10,000 base pair. But the DNA polymerase go back and read again and proofread the protein happened or rising, and in this time, after a proofread the correct mistake, also the mistakes may be still, but this mistake, it is be very, very, very little. It be about 1 mistake or one mistake DNA or 1 mistake nucleotide for a billion base pair error. Here, I think you see this pictures previous, and here the 2 strands of the DNA, and one of them when be replicate, one of them become correct strand, and the others a mutant happen. External influences can create mutations. Here, mutations can also be caused by exposed to specific chemicals or radiation. As I mentioned before, these agents cause the DNA to break down the DNA. When the cell repairs the DNA, it may not do a perfect job of the repair. It not correct well due to the radiation or due to the chemicals. So the cell would have DNA slightly different from the original DNA. And hence, or in this time, a mutation occurs. So that the DNA polymerase cannot correct all changes happen in the DNA or in the new protein, so that it be referred to the new mutation or become a mutant occurs. Here we have in this picture the radiation, chemicals, and infection agents. Here, infection agents, sometimes the virus make the mutation, and some bacteria, I give you two examples for them. This is also make make a mutation, but in what? In the next generation, not in the affected person, but in the next generation for them. The radiation make. Also the X-ray, when anyone working in the X-ray may be exposed to the mutation for their or for her or his generation. And UV also make the mutation. And as you know, the chemicals or using currently chemicals may be also due to or make the mutation. Here, the briefly about, or this is the briefly about the mutations, and for the next lectures, I will complete the part 2. As you know, the Google Zoom have only 40 time or 40 minutes, not more. And this is make problems sometimes, so that I cannot complete more because it was cutting in a few minutes. I will put this lectures on the model, and you can, if you have any questions, you can ask me later or put it your questions on the model. No. Kokia, ظني أفشي وزا أفوضع بيجاكي بلوفوجيا حتى إن شاء الله درست بيت هذا في شكل بيت درستها محاضرة وكيما بس موني تشبكين موني أفشو لا يهوصونا أفاتشي درست شنو يهكون بيجين سباسي هوات كم وهكا يعني هوا بسيارة كبيت أزي حوزرم همي 2 دقائق موين 2 دقائق 3 دقائق موين هكا يكي تشتاك هبيت بلو بوم بيجيت بلازجين كده أبخو قطع بيت نازي قطع كم
درست دكتور جالاكسي باز بوده ما بخوش بسيارين بجاقة جامعات سكنة.
بله سپاس مريم.
دكتور بس بزحمت محاضرة عملي دي چه وقت والگرين؟
ام بخو برای خده جدول من يعني تيك تشوش برشا مكان كين جوارجيت ما ساعات ايك وارجيت حتى سي روجو سي شنبي ونينا ما باصو وارجيت برينه بس درسك بمزيدة بو يو صفاول يو تحليلاته جو بيت من روجو سي شنبي ركنت كتبون هكا مدونه بو سي شنبي چينات بو ينو متشكر من سه شنبه لغو کر من از هات ما 4 شنبه یعنی از شنبه و 1 شنبه و 4 شنبه از حاضرم فون سه روزا جایا هوا چون محاضره نیوکه یعنی مثلا از نهی حتی 11 او محاضره بجنیو ایکیه محاضره 2 یا هوا ببیت الساعات 11 حتی 1 بجو هوا ساعات 11 دوید یا ساعت 12 دوید اوجی درسته و صبحی تمام دیو جدولی تا قل من هيك زردشت يون هينشون
تمام يا دكتور.
تمام
أنا درست دكتور.
بجا عندك ولو وقت بيجاك يعني أبديها بدفت استفادة جيب كان شترة شنو أظن عندك مثلا احتمالات مجيلة عندك هم يد نوف جهاد واشتنا ربوينا مني أف شو مش ميجا يوكلي وبسلمة هوتي بينه بس على كل حال دفتر وخفت انه يجي دفتر يبقوا جاي قريته أبد إنه قبوت بيت وهكي
بس عفو دکتۆر
بله فرمو
بەس هەگەر تا پێشوەخت بکوت بۆ زەرەدەشتی دە ئەمزانین کەنگی بێنە عملي
همو هو قوي همو دي بيش ما هو صباحي على أساس ساعات يوزضايا بس من محاضرة سبيديو هاي من سبيديو هاي محاضرة هيك بلو بيشك استراحة بين محاضرة ومحاضرة دي هبيت هم دي دست بكين بدل يوزد دي كين الضوزده
بس
درست
دكتور عفو
بله
ما ساعدناه محاضرات أصبعي
نا هينينا
بل النهاية
نا المحاضرين سعد يو إنا
ما
نيدا
ساعدناه يوتا دكتور ساعة يوزده سيده هو كيرا بادي فلود معي
نا يو هوت يا جهرين يهوت يا جهرين از دينه بيجمع أبي توشي بيجا يعني رئيس قسم يجي أجدي بيجمع يو چنكي مبخو ساعات نه يو هاي حتى يوزده اديال جماعة الصيدلة بشتي هنجيو هوايا بشتي ويه جورك وديو صيدلية بس ما ترتيب كربو احتماله وانا قهرية أبي ترتيب اديال جماعة أبي يعني هشتو ما يموي حتى صباحي
تمام
قال وان ترتيب كي يهوصون دو جدول ما بجنجي تبع يعني هكا ساعدناه بيت يعني معناه يو تجينها يو صيدلة جينها ما ولك ما بوشي جدول قهري قال السيدة يهوجل كده أو بين يعني دو بومجي تشبيت أبيت شو مشكلة نينا أجي هالوقت بيت درست وشكرا